Okay, good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. It is a great pleasure to welcome you to this sharing session with topic Introduction to Viral Metagenomic Analysis, held by Research Center for Applied Microbiology, National Research and Innovation Agency BRIN, today, Monday, June 3rd, 2024. In this occasion, we are honored to welcome Dr. Ahmad Fathoni M.Eng, Head of Research Center for Applied Microbiology BRIN. Our speaker, Dr. Callum Le Lay, researcher from CSIRO Australian Centre for Disease Preparedness, ACDP. And our moderator for today, Sugiyono Saputra S.Si Ph.D, researcher from Research Center for Applied Microbiology BRIN. Ladies and gentlemen, next is opening remark from Head of Research Center for Applied Microbiology BRIN, Dr. Ahmad Fathoni. Time is yours.
Okay, thank you, Miss Lucy. Can you hear my voice?
Yes, okay.
Yeah, clear? Right, thank you. Okay, good morning, everyone. Honorable Speaker, our guest from Australia. So he came last week, Dr. Callum Le Lay, and all participants, dear researchers and students. Assalamualaikum warahmatullahi wabarakatuh. Good morning. First of all, I would like to thank you, Dr. Callum, for your time to be here in Cibinong with us, and also everyone who has joined this webinar. So today we have one speaker, and so he will talk about viral metagenomic analysis. So I think this is very interesting topic that we are going to have also a workshop this afternoon, but for limited participants, I guess. So I request to the committee that this morning session is for everyone, so we can gain benefit from our speaker here today. And so this webinar and also this activity is a collaboration between Research Center for Microbiology led by Dr. Sugiyono Saputra and his collaborators from CSIRO. And like last week we had a webinar with Dr. Patrick, and today we have another webinar also from CSIRO, which will be Dr. Callum Le Lay. Okay, everyone. So hope you enjoy this webinar, and I hope that today we will have a fruitful discussion, and also this afternoon for the workshop. Yeah. Okay. I think this is all from me. Thank you very much, everyone. And again, Dr. Callum, thank you for your time and also previous knowledge. And I officially open this webinar. Thank you, everyone. So I gave the screen back to Ms. Lucy. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Fathoni. Ladies and gentlemen, before we start the agenda, we will have a photo session first to all participants. Please activate your camera and for the committee to take the screenshot. Okay, I will start. Come from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat feature. Okay, now we are going to start our discussion for today, which will be led by the Sugiyono Saputra, S.Si Ph.D. To Mr. Sugiyono, time is yours.
Yep. Thank you, Ms. Lucy, for your brief introduction to this webinar, and also thank you for partnering for opening this sharing session. And is my voice okay? Everyone can hear me well?
Yes, clear.
Okay, good. All right. Thank you everyone for joining this sharing session. So today we have a special guest who actually have been here since last week. And apologize, could not attend in person with you, Callum, but I'll be there in the afternoon today. And let me introduce myself. My name is Sugiyono Saputra, or Sugi. I'll be your moderator for today's sharing session. And today's topic is a bit unique, and probably some of you may have heard about microbiome, or maybe bacterial community, or 16S, the sequencing. And that's maybe more familiar other than phyromics or microbiome or metagenomics. And this topic is something that we are currently learning as well with our colleague from ACDP, CSIRO Australia. And today is a great opportunity for us to have a brief introduction about the phyromics or metagenomics, which you may guess that this is like metagenomic sequencing, but we'll be focusing on to get the any virus available in that environment sample. And I'd like to introduce our speaker today. He's Dr. Callum Le Lay. He's a research fellow in the Australian Centre for Disease Preparedness, or ACDP, Australia. He got his Ph.D. on discovery of divergent RNA viruses from the University of Sydney, Australia in 2022. And his background is actually computer science and genetics. So he's probably more working with dry lab or analysis or bioinformatics of genomic data. His bachelor degree obtained from University of Queensland, Australia in 2017. And he has several publications related to RNA viruses. And I think, yeah, it's a good opportunity to us to have more understanding on viral or virus from environmental samples, including from from animals, wildlife that we have been working for, and also from like water, soil, or any environmental samples. All right. If you find any missing gap or questions or anything you want to ask, feel free. Just drop your questions to the chat box, or maybe you can raise your hand and in a discussion session after the presentation from Callum. And would like to Callum, are you ready?
Yes. I hope so. Can people hear me through the mic? Yeah.
Yeah. Okay, I can hear you well.
All
All
right.
right.
I'll start setting up.
Okay.
Thank you, Sugi.
All right, Callum, I think it's over to you. Just to give us a brief introduction about yourself and also your topic for today's talk. Callum, over to you. Thank you.
Thank you. Give me a moment. Let's do this and that. Okay. Let me selamat pagi. Matern suwun. We're gonna do this. Hatur nuhun. And then assalamu alaikum. Yeah. Saya suka bahasa Indonesia banyak. Let me just rearrange this a little bit more. I'm unmuted again. Okay. Hello, my name is Callum. Nama saya Callum. I tend to speak pretty fast in my presentations, but I'm going to consciously try and go slower so that I don't overwhelm you with the English. But my name is Callum. I'll be delivering a presentation to you today on virus metagenomics. The talk is sort of in two parts. So the first part will be an introduction and a bit of explanation on RNA virus metagenomics. Some of you may already know parts of this. I wasn't sure what knowledge the crowd has, but I think maybe a lot of people know more about bacteria than even I do. But I will do an introduction on viral metagenomics, and then at the end I will talk about some of the projects that I worked on in my PhD. So to start, I am from the international team within the Australian Centre for Disease Preparedness at CSIRO. So that's a lot of names to remember. You guys can just remember CSIRO. And I live and work in Geelong, in Victoria. So I should actually at this point probably acknowledge the Wurundjeri people, the custodians of the land that we work on in Geelong. And the reason I'm even in Indonesia is that, oh, that squished it. Oh, well. I am part of the WIVRIDS project. So the WIVRIDS project is the Wildlife Interface for Omic Regional EID Surveillance Program, EID being Emerging Infectious Diseases. The acronym is long. WIVRIDS is a long acronym, but it's meant to be like Vivrids or Viverridae, which is that very squished picture that we can't actually see anymore. But these are small mammals that Frank thought are commonly have been host to or reservoirs for unknown emerging infectious diseases. So this project is led by Frank Wong, and the aim is to build sustainable surveillance programs within Southeast Asia. So we're operating in Indonesia, Cambodia, Philippines, and Laos. And the best section of our project are in Indonesia and Sugi's team that I work with, because they're brilliant. The aims of the surveillance program is not exactly to predict new emerging infectious diseases and not exactly prevent, but to prepare. So that is setting up systems so that we can know what viruses could potentially be emerging infectious diseases and know what hosts could be playing a part in it, but not as much predicting if it'll be the new one. It's giving us a base so that we're more prepared for when an emerging infectious virus arises. Hopefully you already know what it is. So in country, in Indonesia, we collect and RNA sequence samples from interface environments. In Indonesia and some of the other countries we work in, this is mostly environmental samples from markets, but we're also looking at domestic farms, hunting villages, bird conservation sites. So sort of any area where humans, domestic animals, and wild animals can come into contact. The most important bit being wild animals. Here in Indonesia, we're collecting anal, oral, and fecal swabs from a range of species carcasses. So we just swab them, don't get any blood or tissue. The idea for this part of the project is broad level collation. So we want to work out what viruses are present really broadly, and identify which hosts have these viruses long term. And then later on, we want to do some analysis of the evolution of these viruses at the markets. But before I worked at CSIRO and I worked in Geelong, and this will be the later part of my talk, I did my PhD from 2018 to 2022 at the University of Sydney. And this group specifically looked at identifying RNA viruses using metatranscriptomics. I myself, during my PhD, was looking at invertebrate and microbial viruses, particularly microbial RNA viruses. So there might be a bit of overlap with some of the bacteriology people, or you might ask me really hard questions that I can't answer because I'm more of a virus and computer science person, but we can try. So I'll be presenting work for my PhD because I think it's fun. And after we get a bit of a grip on the viral metagenomics, I'm hoping it'll be easier to understand. To get started, Oh, it's really basic. I reckon a lot of people are going to know this already. But the central dogma of all life, of all biology, is you have DNA, which is your information storage within all life form, and this has got encoded in it everything that we need to build me or a bat or E. coli. RNA is well, RNA actually has a whole bunch of different uses within life, but the most well-known use is that it is used to make temporary transcriptions of parts of the DNA, which are transported to the ribosomes, and then we produce proteins. I do think a lot of people, I reckon a lot of people know this already, but just to make sure we're all on the same page, I thought I'd go through that just briefly. The important bit is we can measure the evolution and identify organisms using the structures of these key molecules. So by looking at the order of the nucleotides in the DNA and the RNA, and even the amino acid sequence in proteins, we can work out exactly what organisms are and exactly how they have evolved, how they differ from each other. We can determine the structures of these molecules using, oh, sorry, my slides are a little bit wonky. I guess because I connected to the computer. Just pretend that's right in the middle. We can work out what the order of the nucleotides or the code of these sequences through sequencing. So at its most basic, sequencing, again, maybe a lot of people know this, but I'm going to go through it. It'll get more complicated later if that's what you want. At its most basic, we destroy the cells of organisms to get the genetic material inside, which is often DNA, but can also be like the transcriptome, the RNA. And then we determine the order of the nucleotides present in the RNA or DNA using a sequencing machine or sequencing platform. Typically, the workflow for this looks a little bit like this. I'm not going to go into detail of this graph, so you don't have to read it. You can just listen to the words I say. The workflow, this is generally an extraction of the genetic material, conversion to DNA if it's RNA, preparation into libraries. So that is sections of normal, all the same size bits of DNA sequences with little tags on the end. So extra nucleotides to help the sequencing machine do its job, but also to identify multiple pools of sample that you submit. Next step is usually there's usually some amplification present in the workflow. It can be at different stages. And that is making lots of copies of the genetic material using PCR, and then the sequencing itself, usually with an Illumina or Oxford Nanopore machine these days, which I got a picture of both there. Let me have a look at that. Is it Illumina? Yeah. So with Illumina sequencing, it's a type of sequencing called short read sequencing. And the way that works is in the sequencing machine, we'll make copies of the DNA that we put in there. And as each nucleotide is added on to a copy, the machine analyzes, I think it's light, it's spectroscopy, as each nucleotide is added. And then we can determine what each nucleotide is in order. Nanopore is a little bit different. They have a protein pore in a membrane, and the genetic material passes through it like a string and a needle head. And as each nucleotide passes through the pore, the pore emits a different electric pulse through the membrane, and sensors can determine what the nucleotide is by looking at that impulse. After that, well, this diagram shows typically the wet lab part of it in a specific way. After getting this done, there's the bioinformatics steps. So we can skip the first two because that's in the bit before, though I think these pictures are kind of nice to visualize it. Get to the sequencing. So in orange is the stuff that's it's not real sequence. This is within your computer. This is your reads. So we typically get lots of short sequences called reads, longer in nanopore, and these are assembled into contigs. So these are very short nucleotide sequences. The assembly works by, you can sort of imagine it as if we have multiple copies of the same book and we tear them apart. Assembly works sort of in that we get bits of the torn apart books, and we match where we can and put them all in an order. So maybe one paragraph matches between two ripped apart copies, and then the later paragraphs stretch onward. So once we've arranged it all together, we can reconstruct what the book's order should be, which is not always perfect. So this analogy, the books are genomes and the reads are the short bits, and it is also not always perfect. Now, this, let me grab my water bottle if I can. Give me a moment. I'm back in business. So this approach is covering a fairly, it's a fairly standard sequencing approach. Metagenomics is what this talk is sort of about. And that is the study of the total genetic material obtained from an environmental sample. So rather than one cell, like you would have in a culture, one type of cell, or like in my analogy, several copies of one book, you take a sample and you destroy all the cells within there. So we might take a swab of this table, a swab of my mouth. We might get some dirt. You take all of the microbes out of that, get all of the genetic material, and then assemble it all at once. So this is much harder because, like in my book analogy, we don't know which bits of which genome belong to actually realistically belong all in one genome. So we have to sort of sort out all the genomes as we assemble. But the advantage is it is culture independent. So that is, we can see exactly all the microbes that are in an environment without having to culture it. And because of that, I think it's been about 15 years since this approach has been used. Don't quote me on that because that could be wrong, but about that. But it's allowed to massively broaden the known biodiversity on the planet, right? So this approach, in some systems like permafrost or soil, we would try and culture bacteria from there or passage viruses, and we would get specific bacteria that can live on gels. But some microbes cannot live without the rest of their community present. They're in a established system, and they're reliant on all the others around them. So this approach allowed us, I think there was about 12 phyla 15, 20 years ago of bacteria, and now there's hundreds and hundreds of phylums. It's massively broadened the amount of biodiversity known on all fronts. Bacteria, archaea, eukaryotes, and viruses. The downside is that if you are looking for viruses specifically, we do not have a way. It's very difficult to say which host the virus is infecting, right? So if we get a whole metagenome and it has a bit of bat blood in there, it has Bartonella, some bacteria, we have some parasites, maybe we have a bit of the insect diet. So we have all of these different organisms mixed in with the sample from, say, like a bat mouth swab. If we find a virus, we can't just say this is a bat virus because it could be any of those organisms that it's infecting. So on that note, let me talk about the virus stuff a little bit. So, some of the words are gone. That's okay. I'll say it out loud. Viruses are obligately parasitic organisms that happen to be the most abundant organisms on the planet. But that is if you believe viruses are actually organisms. I guess there's a bit of a debate there, but I'm not gonna, if you ask me that question, we can talk about it after. I've worked with RNA viruses specifically. So that is viruses that have an RNA-based genome as opposed to a DNA-based genome. The characteristics of these viruses are that they have a much, apart from having RNA genomes, is that the genomes are quite small relatively. So a virus genome can be, RNA virus genome can go from about 2,000 nucleotides to about 30,000 nucleotides. I think I'm giving that a pretty broad range. So the genomes are quite small. What if I can, there it is. They also have low fidelity polymerases. So what this means is the proteins that they make that make copies of their genome are kind of bad at the job. So RNA-dependent RNA polymerase and retrotranscriptase are the most common RNA polymerases in RNA viruses. I'd struggle to even name another one, actually. So I think they're pretty ubiquitous within the RNA viruses. But these introduce a lot of error. So one mutation per every replication of an RNA virus. And the third characteristic is they replicate a lot. They replicate a bunch. Viruses can make lots and lots of copies when they're infecting someone. Their RNA polymerases makes lots of mistakes in that genome, in the copies of the genome. And what this means, the synergy, the mix of these characteristics, mean that, can we see it? Oh yeah, there's a little picture. That in a pretty short time, and I say short, like, millions of years, but short for evolutionary history, we can change all of the sequence within an RNA virus genome, or a large majority of it, right? All of the genetic material of 5,000 nucleotides can change in a relatively short time. And what this has led to, and this tree figure here, you don't have to understand the details. It's just that if you've ever seen a tree of all life, so bacteria, archaea, and eukaryotes, you would see it's all one tree, which is kind of neat. But with RNA viruses, they're so different. We can't build a united tree, and it's also in question whether they form one big tree, even if that is actually true. But the reason for that, the reason that we have all these different trees and not one big tree, we don't know how these families, classes, phylums interrelate, is because of the characteristics of the RNA virus. So this is a bit of a summarization of the difficulties with this research, because I've mentioned a couple before. But first, it's hard to match hosts in a metagenome or metatranscriptome. Sorry, I'll use metagenome and metatranscriptome sort of as the same thing, because even though metatranscriptome usually means RNA, I'll say metagenome because I'm looking for genomes, like a metagenome. So I'm sorry if that'll be a little bit confusing, but we're going to stick with metagenome, which is all the genetic material from a microbiome. So the first thing is it's hard to match hosts of viruses to the actual viruses through metagenomic data. It's also difficult to match genomes with themselves. So if we have multiple segments of a genome, or it's we only were able to sequence it in 2 bits, or there's multiple viruses of the same, like multiple related viruses in the same organism, it can be really hard to sort out which bits of which genome belong to which virus. Next, we find, so once we have all our data together, we, wonder if I can put my mouse on this screen. Nah, I can't, I'll just have to describe it. When we're trying to detect viruses, we use similarity. So we use all the existing virus sequence and all the existing bacterial sequence to find, to detect organisms in our sample. The problem with this is if the organism is too different from stuff we already know, we have a lot of trouble identifying it. And of course, as I said, with RNA viruses, they change so quickly evolutionarily that we have part of the world virus sphere, RNA viruses out there, that we have a lot of trouble and maybe cannot even identify currently because they're just too different from what we've ever seen. So this picture of a tree here, the idea of this is that it's talking about the entire virus diversity on the planet, the entire virus sphere. Orange is stuff that we can detect with metagenomics, so sequence that we can obtain with metagenomics. The blue is what we can detect with PCR, and red is what we can collect with virus culture. So we can look at viruses that are more and more different from what we know using metagenomics. But the problem is we can't understand everything. the further and further we get away from stuff we've actually cultured, stuff that we have PCR for, stuff that we have existing genomes for, the harder and harder it is to say this virus is a real virus, or what even is this virus. Other hurdles are, there is a fair bit of misannotation within the existing data. So the field has grown so big, people are looking at lots and lots of viruses, lots and lots of sequences. And like I said, some of them are so different. There ends up being a bit of misannotation. Yeah, right. Now, we've had a bit of a lecture about how virus metagenomics might work. So the rest of the talk is I'm going to discuss some of the projects that I worked on during my PhD, again, because I think they're kind of fun. And maybe we'll see if this first part helps us understand it a little better. So termites, I want to talk about termites first. In 2016, it's a very small box in that right-hand corner. But in 2016, a researcher, M.Eng, who was in the lab that I was in my PhD, found 1,600 novel viruses. I have to check the paper to get that number exactly correct, but 1,000-ish, 1,600-ish novel viruses by looking at a huge number of arthropod samples from China. So this is actually like a massive study. And at the time, this was a lot more viruses than anybody had found at once, though it's quickly increased. We're finding more and more with bigger scale projects all the time. But at the time, back in 2016, I guess the most viruses that people would find in a study, novel species of virus, would be about at most 200. So off the back of this redefining the invertebrate RNA virusphere paper, I wanted to look at both RNA viruses, look at the RNA viruses of termites, and also hopefully their gut symbionts. So the goal was it's fundamental research. We want to find more and more viruses because, again, the more different viruses you find, you can use those genomes to look for even further viruses because they give us like an anchor, something more similar to look with. So termites are insects in the family Blattatodia, which I think they also share with cockroaches, interestingly. They are social insects, so they form large colonies, and in the higher termites, they form like quite complicated mounds, termite mounds. My interest with them was a little bit because of their gut microbiome. So termites have a very specialized gut microbiome that they use for metabolizing their diet of roots and grass and soil. And if you were to take that gut microbiome away, they wouldn't be able to live. So not all organisms are like this, but termites are dependent on the bacteria, on the microbes that live within their gut. So I thought it would be interesting, though I probably made my life hard, but I thought it'd be interesting to look at these termites, because we know there's lots of viruses in insects. Look at these termites, but also look at the gut microbiome in there at the same time and see if I can find a bunch of viruses and work out what they are. So I collected 54 samples of 50 species of termite. The majority of the samples were pooled. So that is, we didn't just extract RNA from a single individual termite. We'd get 10 or more individuals with a bit of a mix of workers and soldiers. Though we did have one set that was separated, like a pool of just soldiers and a pool of just workers. Importantly, some of the samples were full body samples, so the whole termite, and some of them were just the head. The whole body samples generally yielded more viruses. So in this figure here, the blue is the whole body samples, and at the bottom half of the figure we can see how much reads we got down the bottom. I think these are, they're not NovaSeq, they're like NextSeq 2000, the sequencing platform. But we got a larger amount of reads from the whole body samples by a bit than the head only samples. But when we looked at the viruses we found, so up the top we have the amount of reads that we mapped to viruses that we'd found. The whole body samples also found a lot more virus, like a higher percentage of virus, though even though the head only did find a couple sporadically. So I identified 67 novel viruses, and these were often about 1% of the reads that we sequenced, which makes sense unless an animal is really, really sick. If you extract all the RNA out, extract the genetic material out of a whole organism, you're going to get lots and lots of the host genome. After that, you'll get plenty of bacteria. And then the smallest fraction, unless this animal is really sick and has lots of virus within its body, that last fraction will be fairly tiny. So less than 1% of these libraries had virus. And of these viruses, we had a lot of diversity in the not termite infecting. So the symbiote bacteria, microbial infecting viruses, we had lots of diversity, but they weren't very abundant. Whereas we got lots of a large amount of reads from the termite specific insect infecting viruses, but not a lot of diversity. Only a couple of different viruses would infect, but at large amounts. So identifying the viruses, I made lots of trees, which I'm not going to show all of them. This one's sort of a summary tree. So these are different, I think they're probably like phylum level, a little bit lower than phylum level groups of viruses in trees. The red groups that I identified new viruses in. So 67 novel viruses, some complete, some partially complete, and all of them were around 30% protein identity, which is pretty different. If we're looking at different kinds of influenza, there'll be 95% at least between them, 95% nucleotide identity. 30% protein identity means that they're quite different. Though later in the talk, we'll probably look at some even more different ones. We could detect these with blasts, but we did have to confirm with other tools to annotate the genome and confirm they're correct. The majority, and like I said, most of the diversity were from the Partiti picoburner. So the Picoburner-Viridae, there was 36 novel viruses in this group, and the Picoburner-Viridae, at the time, and I think still now, there's not a very well-described host. People aren't absolutely sure what the Picoburner-Viridae are actually infecting. I think the lack of study on this is because it's not making humans sick anytime soon, so no one is too worried about it, but they're highly associated, I found, with feces samples. So wastewater, some people who, any kind of like fecal samples, you might find a fair bit of Picoburner on. And these are quite small genomes. My suspicion is they're microbial infecting. So fungi, yeast, or bacteria probably. The other group that I found a lot of is the Nana levi. So this group is called the Lenarviricota phylum now. And I say now because they change a lot of the names within these families quite often, which is a bit confusing. But I found 23 viruses in this group. And again, these are very small genomes. I think they're only like 1,500 nucleotides. And this group is one of the only RNA virus groups that are known to infect bacteria. So they have viruses in there that we've confirmed infect bacteria. Apart from these major groups, we found a less diverse range of insect infecting viruses. And this is from the Phasmaviridae, Polycipiviridae, Eflaviviridae, and the Flaviviridae, which I'll talk about in a moment. Oh, I should also say the Lenarviricota phylum also infect fungus, or the mitochondria of fungus, which is kind of interesting. So a lot of the viruses we believe are the termite gut component. These are viruses that are infecting the termite gut. Let me get a bit more water. I hope I'm not speaking too fast, but let's see how slow I can go. Sorry, I get nervous and then I speak faster and faster, but that's pretty hard when you're not a native English speaker. So the insect infecting viruses, Phasmaviridae, Polycipiviridae, Flaviviridae, are all known insect infecting viruses, but the flavies that we found were pretty weird. So flaviviruses are actually well known publicly. You definitely know the name of different flaviviruses. West Nile, dengue, yellow fever, Zika. and a lot of other tick-borne viruses that you might not know the name of, but they're pretty common. If you're getting sick from ticks, that is one of the things that can cause it. It was really weird, without even like looking too much at the genome or anything else. Just finding a flavivirus in a termite was pretty weird, particularly because these viruses are vectored. So that means either a tick or a mosquito or other blood-feeding insects are usually part of the life cycle for the flaviviruses. So to find it in a termite is really unusual because termites aren't biting mammals. They can't really be a vector, and it would be unusual for, or unusual at the time to find a virus, a flavivirus that infects an actual insect. So an insect-specific flavivirus. So this tree here is the Flaviviridae, which is the family. And we can see some very small clustered trees with labels, and those are the known flavivirus genuses. And this big tree out here with the red labeled A is where I found the flavivirus in termites within, which I called Waxystomes. And this bit of the tree looks so big because the distances, so the evolutionary distances within this bit of the tree is quite long. And the group we found it in was very, very weird. So the closest relative to Waxystomes was a squid virus, which had been recently found. They found similar viruses in fish and shark flavivirus. This tree actually has a lot more members now, so it's a little bit out of date. I'll say some of the other ones as well. But within this group, there's also like a bat. So we have bat, fish, squid, shark, termite. And now at present, we even have, I think geckos are closest to the bat, RNA viruses in geckos. And we have some frog RNA viruses, I think, within this group as well, and some other insects. So at the time, we thought, this is bizarre that we're getting a bat, then a fish, and then termites. Initially, we thought maybe it's not exactly infecting these organisms themselves. So it's, like I said, you can have lots of different organisms within a metagenome. Maybe these viruses are all infecting some sort of parasite. But with the shark virus, they ended up finding like an immune response, RNAi, for the flavivirus. So it's likely that it was actually causing infection, at least in that case. So all around really odd, different, very broad range of hosts, not usually a vectored kind of organism. And the other thing I wanted to say about this group was, what was it? I can't remember. I think I do remember. It's a lot. It's indicative that it's this broad range of hosts is, to me, I thought once we'd sort of ruled out some other hypothesis, that this group was a really big group. This could be like an order level group, but at the moment we'd only sampled like 3 different viruses within it, which is, yeah, kind of cool. It's greatly expanded now. They found the semen flavivirus within this group, and the people who found the semen flavivirus called it Phenoptera, which is Latin for fins and wings, but that was before they found frogs and geckos in there, so the name didn't really stick. So zooming back out now. Sorry, I wanted to talk about the flavivirus because I think it's kind of cool. I mentioned some viruses that I said were termite infecting, or I said were microbe infecting. But like I said, the host matching is actually one of the most difficult things. How did I do it? I just did my best with the information that I had. And even then, it's sort of just an inference. If we really want to confirm hosts for viruses in these organisms, you have to isolate them. But it's good to give an inference, because if someone finds another one of those flaviviruses, they kind of want to know what I think the host is, what my best guess is. So this chart combines 3 things: the abundance, which is on the y-axis, the identity to the closest relative, so that is how similar it is to the closest relative. then the shape of the dots on this plot are what the closest relative is known to infect. So a combination of host and how similar they are to the closest relative, how abundant they are, and then also whether they were found in the head only samples or the whole body samples was sort of how I determined which viruses I thought were termite infecting or microbe infecting. So like I said, the large diversity of it was these microbial infecting, but then these clusters up top, I believe, were the insect infecting viruses. So they're usually more abundant, have relatives that are known to be insect infecting, sort of like the flaviviruses. And we found some of these in just the head only samples, so only in the head. So there wouldn't be as much, there's still like the oral microbiome, and there's definitely like still a fair amount of bacteria there, but without the gut, the large symbiont mass, it gives us better odds that these are insect infecting viruses. All right, so this gave me, my final list of the different viruses within the different groups and their identities. So yeah, around 30% protein identity. And I was able to sort of allocate them to whether I think they're symbiont or termite infecting. The way I see here, I also had some that I thought were maybe plant, or some that I couldn't say with a lot of certainty what I thought they actually were. Next. So I hope it's not too big a jump. I can sort of explain the logic between the different between the different projects. I had to explain the logic in my thesis for my PhD. So to make it make sense, I wanted to find really different looking viruses, so really divergent ones. That's what I did in my PhD, was expand diversity, which is fundamental research. I looked at invertebrates and the symbionts because there wasn't a whole lot of bacterial RNA viruses identified at the time, prokaryotic RNA viruses, even though that word is not really a great word. So I was sort of prompted to look at the termites because I wanted to gut microbiome. The idea with geothermal pools is that I wanted to look at these sort of very different ancient bacterial groups and see if I could find RNA viruses in them. So I sampled from actually quite a lot of different pools in New Zealand, and these varied greatly in temperature and clay content. I sampled from a lot, but in the end, I only got 2 pools that I could get any sequencing data from. So I had one pool, which I think is K8. You don't have to remember the names, but this was a 71.8 degree pool, so very, very hot. And 98.5% of this pool was archaea. So the microbiome, or the microbial community within there, mostly large majority archaea. My other pool was less hot, 36.8 degrees, 58% bacteria. So there was like far more diversity of microbes in there. There's still an archaeal component, but quite small. So the hotter pool in particular, but these hot springs were closer to the conditions of early Earth, like 3 billion years ago. High temperatures, a lot of sulfites, and some of these lineages are sort of have been sitting and evolving in these hydrothermal pools and hydrothermal vents for a really long time. It's very selective what organisms can live in these high temperatures. So the archaea in the hotter pool were majority the genus Sulfolobus, and in the cooler pool we had a bit of a mix. So what viruses did we find? This is a crappy slide. Sorry for that. It's just a big old table. I identified 23 novel viruses within this group, mostly from within the Lenarviricota. So this is that group that I said are probably the only, within this phylum, the only viruses that we sort of know, apart from one double-stranded RNA viruses. Within this group, these are the ones we are pretty sure are bacterial infecting. I found 23 viruses within them, but specifically within the Mitoviridae and the Sobelli viralis. But the bizarre thing, so I'll say that some of these similar identity to the viruses that I found in termites, because there had been some other projects looking at aquatic viruses that allowed them to be pretty similar to the viruses that I found. But the bizarre bit is I only found them from the cooler pool. So in my very hot pool, I didn't find any of these viruses at all. So some of the interesting viruses that I found were these guys within the Lanao varicoda. These sat in phylogenetic positions with not much taxonomic definition and were basal to the Cryptoviralis and the Fias viridae. What I'm getting at here is it's similar to that flavivirus bit of the tree. As in, I found viruses that were sitting in locations where they, if we sampled really, really highly and we managed to go look at different viruses in a whole all over the planet, these groups would expand to be like order level clades, I believe. Like these are very few viruses have been found, but the scale of the taxonomic groups is quite huge. And the interesting bit with some of these viruses is it once these were present in the tree, it sort of rearranged some of the ways that the different virus clades were sitting within the tree. Though this wasn't unusual. With this Lanao varicoda phylum, we'd found that the position of the tree could twist around and change quite a lot as soon as you add a different organism. Some of them were quite divergent, these viruses, while others were added to groups with a couple of existing members, like I said before. Okay, so coming back to finding no RNA viruses in that hot pool. Why? So an interesting theory, well, an interesting bit about archaea RNA viruses is there isn't any. So we found no confirmed, validated RNA viruses that infect archaea at all, which is really bizarre. I'll show you a tree in a sec, but like archaea are a whole kingdom of life, you know, equivalent to bacteria, eukaryotes, every organism we can see. Archaea are a group in the evolutionary history equivalent to that, and we find no viruses in them. And this could be undersampling, because a lot of these groups are not, you don't get sick from archaea. And if archaea gets sick, we don't really care too much. So it's it could be undersampling that we don't see any RNA viruses there, but also RNA viruses infect pretty extreme environments. I'll talk about that a little bit in a sec once I get into the further slides. There had been a single RNA virus study of geothermal pools, similar to the study that I had done before at Yellowstone in America. They identified some viral contigs, but later it seems that based on the evidence, these virus contigs, these genomes that they'd found, partial genomes they've found, were probably eukaryote infecting based on the relatives and the codon usage. So there's a specific codon usage that they believed would be compatible with the archaea living in the hot springs that they talked about. Anyway, so I'll briefly go over the archaea themselves. So archaea are this third kingdom of life. 3.5 billion years ago, bacteria and archaea emerged under early Earth conditions. This tree is not rooted. Let me see if there's another one. Look at this tiny little tree here. Look at that. 3.5 billion years ago, where it's labeled as Leuca, the bacteria and archaea split into two groups. And then a short while, and I say short, this is in millions and millions of years, the archaea and eukaryotes split into two branches. So that's 3 kingdoms of life, but there's a little bit of an order to them. I'm talking about bacteria, and I think there's a lot of bacteria people here at BRIN. So you guys might actually know way more than me, but you're going to have to listen to me anyway. The archaea split I believe is due to an well, the theory is that it's due to an endosymbiotic event. So that microbes that moved into the archaea caused a divergence between the archaea and the eukaryotes. Let me go back to this slide now. So the thing about archaea is you don't see them everywhere. Well, you can't see bacteria either, but they're not present all over the place. A lot of the conditions that archaea live in are sort of like early Earth, 3.5 billion years ago conditions, similar to back then. They're very extreme. So they're often present in higher levels where bacteria cannot live. So hypersaline lakes, really salty lakes, gula, no, is it gula? Sorry, garum. Anoxic environments, so very little oxygen, and acidic environments where there's lots of acid, so acid mine drainage is one, and very high temperature environments. Aside from that, they're also found in soil and guts a little bit, specifically a group called the methanogens. So these are archaea that perform a specific type of metabolism. They metabolize methane. So while there's it's not exactly relevant to my talk, but I think they're really cool. While there's no RNA viruses found in archaea, there's a whole bunch of different DNA viruses found that infect archaea, and they have really weird morphology. In the top left corner, I think is some of the Cordoviricites. And these are those like moonlander looking viruses that you might have seen before. And these actually infect both archaea and bacteria, which is kind of crazy because you're bridging the gap between two kingdoms. But these viruses infect both. Some of these other viruses are archaea specific, but they have really weird morphology. So some of them are quite squishy, which is like the Pleolippoviridae. Some are long and filamentous. Some are even shaped sort of like vases. So they're like a really interesting morphology of DNA viruses that infect archaea. They're really interesting. And I know research with the DNA viruses in archaea, there's a lot of horizontal genetic transmission. So a lot of changing of genes between the DNA viruses and the archaea, which is really cool. So this last project this is the last one I'll talk about, then we can have some discussion. This last project was aimed at specifically identifying RNA virus genomes associated with the archaea. Actually, a lot of the projects that I'd done before this, they didn't find RNA viruses in archaea, but they were sort of related to finding RNA viruses in archaea, because I know there was archaea methanogens in termites. I know there's lots of archaea in the hot springs, but this last project was we don't care about the bacteria. We don't care about anything else. We want to find viruses in archaea and know for certain that this is an RNA virus infecting archaea. So what I did, well, the idea is, like I said with metagenomics, if you get a culture independent sample, you get more microbes. And this is sort of true in viruses as well. If you take an environmental sample, you get more diversity. But then the problem is it's hard to say what host the virus is actually infecting. And in an environmental sample, I can find environments where there's mostly archaea, but there's usually still a little bit of bacteria, a little bit of eukaryotes. And if you find an RNA virus in there and you publish it, people would go, is it not infecting the bacteria? Is it not infecting the eukaryotes? You have to prove it, which is really hard to do when you just have data. The other aspect is you can get cultures of archaea. And then if they're pretty pure cultures and I find an RNA virus, it's perfect. I can go, this is an RNA virus infecting archaea. But the problem is you don't get the diversity. You don't see as much viruses in cultures of organisms. So the logic was I was going to do both. So first I sampled, well, not chronologically, I sort of did these both at the same time, but first I sampled from a salt lake in Victoria called Lake Torrell, and that's what we have pictures of. I would pump liters of water onto 0.2 micron filters. So Sugi said before I'm bioinformatics, which is true, but I've also done a fair bit of field work and lab work. I'm just not very good at it. But I would pump liters and liters of water onto these 0.2 micron filters. And these were small enough to, well, I also pumped them through glass filters as well, but these are small enough to catch the bacteria, like the microbes on the filter. And it was really easy to see when I built up a fair amount of microbe on my filters because it get really pink. I should have included some of the videos of it. So I got this hypersaline water, really, really salty, pumped it onto a filter. It got this pink color because of the keratin. So I could tell there was lots of biomass on it. And I extracted that hoping to get mostly archaea, because I know in a previous paper I had looked at the microbiome of this lake, and because it was fairly nearby, it was a little bit of a flight from Sydney, but I could actually go there with my pump. But from a previous study, they'd found that the large majority of the microbes within these salt lakes were archaea. So that's what I was aiming for. The second part of this project I extracted from cultures. So there was a group in Sydney, Rick Caviccioli, and there's other groups there that look at archaea and DNA viruses in archaea, but they had cultures of archaea that were seeded from lakes in Antarctica. And these were hypersaline lakes as well. So these are a bit different from the ones that I looked at environmentally, but the thing was these were enrichments, these were cultures, so they're way more, well, the idea was there'd be more pure archaea, and if I find a virus in them, great. So these samples themselves looked pretty good, really. Like my environmental samples, my my enrichment cultures all looked majority archaea. So more than 70% for all of them. Again, it's not ideal. Like I would like 100% so that I don't have to, if I found an RNA virus, I'd be, I could go, yeah, this is definitely the host, but you work with what you can. And I thought this was quite good for an environmental sample to have upwards from 70% of one organism. The archaea specifically was a couple of different strains of Haloquadratum walsibii, which I had a little picture of before. It's this green one here. They call it quadratum because quad is square in Latin. I don't actually know if they're square or not. People have told me that they only looked square because when they pressed them on the slide that they looked that way. But that green, those green cells there were the original pictures of Haloquadratum walsibii, the archaea that are in the salt lakes in Lake Tyrrell. Yeah, so the hosts are what I was looking for. I blasted these samples, found lots of DNA viruses, but absolutely no RNA viruses with just blast. You'll probably think I'm going to find some eventually. This is a sad story. This is a spoiler. I don't end up finding any RNA viruses in the archaea, but I do a whole bunch of stuff to try and find it. A lot of it was removing sequence that I know is not virus, so that I can really narrow down the groups of sequence left that I know could be virus because they're not anything else. And then I used a whole bunch of tools. I used different types of hammer. So that's hidden Markov model searching tools. And I use that with a range of databases. I use tools like PalmScan, which use position-specific matrices to try and detect motifs of the RNA polymerase. I used some structural predictors. I will say this is before AlphaFold. So AlphaFold could be really good for this now. And I was thinking I should go back and, try and run AlphaFold, but it's a bit tricky. I used Fire2, which is a structural predictor, to try and predict the structure of some of the random sequences that I had, and then see if this structure is similar to virus capsid or virus polymerase. I did these smaller and smaller sets, and I gave them scores. Maybe if I go over here, I sort of scored them based different sequences on how much I could think that these might be real RNA viruses. But it's all in all sort of a negative result, because even though I could find some contigs that I thought were the best of the lot that I looked at, none of them I thought were really believably real. So from this study, I think, oh, sorry, I should also say I did dinucleotide analysis. So that is looking at how common certain sets of nucleotide pairs are. I'll have to explain the idea here. So there's a similar thing as codon analysis, which is sets of 3 nucleotides. Dinucleotide analysis is sets of 2. The idea is viruses reflect their host genomes a little bit. So you want your genome to look a little bit like your host genome, have similar characteristics to your host genome, so that the ribosomes and the machinery within your host genome replicate you effectively. So there's a bit of a bias in the dinucleotide frequencies. I should have got some of the other pictures of this. The dinucleotide frequencies to be more similar between viruses and their hosts. So this is pretty distinct for viruses that infect plants and viruses that infect mammals. And that then it gets pretty fuzzy when you're getting to very specifics. But I thought this would be a good way to go about it, because archaea are a whole kingdom of life. Maybe I can match the dinucleotide patterns there. So this was part of what informed my scoring of top candidates later on. But unfortunately, with this study, I ended up not finding, I don't believe I could conclusively say I found an RNA virus in this group. I had some top candidates. Right at the end, when I was publishing my study, I used some AI tools that had just come out, not AlphaFold, but an AI tool that was made for detecting viruses. But the problem is, and I think I spoke about this before, genomes that are this different, less than 20% protein identity, you can't really tell what they are. You get sequences, maybe an AI tool identifies them, or I get a good scoring, but you look at this sequence and you go, how do I annotate this? Where is the genome, the coding sequences within it? Is this even real? It's too far out from what we know to really be conclusive. But even though it's a negative result, I thought it was kind of interesting that, I could try this hard and not find any RNA viruses for a whole kingdom of life in these pretty pure samples. I mean, it would be worse if I found some RNA viruses, but they were bacterial eukaryotes. I found no RNA viruses in some of those hot springs that were entirely archaea. And I found no viruses from this study in looking at these hypersaline lakes. But at both the hot springs and the salt lakes, we found lots and lots of DNA viruses. DNA viruses for archaea seem to be really common. There's some caveats to that. Some of the things that could have caused problems is the extreme nature of these environments can interfere with the sequencing and the library prep. Maybe the extreme nature of these environments stop RNA viruses being able to move around, which I don't quite believe, because I think we have extreme environments where I do find RNA viruses. But it could be the case that RNA is just not very stable, and maybe even with the protection of a capsid, it just doesn't move very well in these extreme environments. But either way, as a negative result, I thought it was pretty interesting to find no RNA viruses for a whole kingdom. But I'm hoping someday someone will, maybe with AlphaFold and some more AI tools, someone will do a better job. And I know some groups are looking to find RNA viruses in archaea still. That's about the end of my talk. Big thanks to Sugi and his team for hosting me, and Pat for the last week or so. Some of the groups that I want to acknowledge as co-authors and stuff within some of the projects that I've spoke about is Meng Shi. He was the guy who found lots of viruses in invertebrates. Nathan Lowe is at University of Sydney, and he helped with the termite stuff. Matthew Stott in New Zealand helped me actually go out and collect samples from very hot springs, which is a lot of fun. The Caviccioli group for their help with the enrichment cultures from those Antarctic hypersaline lakes, and the Holmes group, so the group that I did my PhD in Sydney. Big thanks to them, because they let me do a really interesting fundamental research for the 4 and a bit years of my PhD. That's me. Thank you for listening. I don't know if I pass it on to someone else, or if anybody, to Sugi, and I'm open to any questions.
All right. Thank you, Callum. Fantastic. And yes, that's a brief, very concise introduction for the viral metagenomics, which is mostly from environmental samples.
Yep.
Which is great. And then I think it's related to many researchers as well, including research for archaebacteria.
Mhm.
And we'd like to invite you, for everyone, in the Zoom meeting or within the room in PNC building. If you have any questions related to what Callum just presented, please raise your hand. Anyone? Any questions? Yep. If not, I'm gonna just read some of questions from the chat box. It's the question from Eki Ilham from Research Center for Fisheries. Callum, can you see this slide?
Yep.
Oh, yep. Okay. And the question is, can we do metagenomics for DNA and RNA viruses together using only RNA polymerase targeting primers? Thank you.
You certainly could. I just wouldn't advise it too much. The problem is there's plenty of RNA virus genome sequencing within the host of most of the organisms, fish for sure.
Mhm.
There's lots of RNA viruses in fish, so it's a good thing to look at actually, and for the Center for Fisheries. But yeah, doing both at once, you'll detect viruses that you might think are real, but they're probably artifacts within the genome. They're endogenous, which is a bit tricky. You could do it. It would just be a lot of work, and I'd probably advise you get the same sample and you use the DNAs on one and you don't use the DNAs on other. The other thing is you'll get so much more DNA. Like the fraction that is virus is quite small, especially in something like fish, where if they're not actively sick, they will just have a number of viruses present. But even if they're sick, I think you might just not get enough reads relative to the amount of host DNA and host RNA to what you're sequencing to actually try and identify viruses. I hope that was a good answer, Eki.
Thanks, Callum. Other questions? Callum, I'm just curious whether how you get the new viruses. I mean, how to consider whether the sequence you got there are new viruses? Anything related to like determine how like the similarity?
Yeah.
Yep.
If it's not
Like,
too different, mostly we'll get the viruses out, we'll try and annotate them, and if they have complete real annotations, then we believe they're more valid. Every virus sort of needs a capsid and an RNA polymerase, so that's what we're looking for most, is complete RNA polymerase and capsid coding sequences. And then in addition to that, we try and place it in trees. So we build trees looking at the relatives and whatever. And if we find that they sit within a position that we can kind of believe, then we sort of believe it's a valid virus. But both of these approaches are dependent on having existing genetic material that is similar to what we're looking at. When you go really, really far away, really divergent viruses, it gets harder and harder to work out if something's real or how it works. I hope did I get that question right, Sugi? Is that was what you were asking? Yeah. You're muted, Sugi.
Sorry.
You're right.
It's mute. Sorry, there's someone who raised his hand from Riza Hartawan, I guess. Please.
Thank you. My name is Riza Hartawan from Research Center for Veterinary Science. I am from research group for zoonotic emerging and remerging disease. This is an interesting presentation about the virusphere in a certain kind of environment. My question is, in the past there is a study that tried to study a virus family in a certain species like bat or in monkey, but I think it's too general to get like a overview about certain kind of virus like a zoonotic disease, because it's too general. Can you give an like a more explanation how we can get the potential of the zoonotic disease of viruses from the very general study like this? And my second question is like when we look like the COVID-19, the virus is evolved from the first outbreak into become very numerous variant. So do you think any specific gene marker or on the virus that can make them survive or become more dominant in the environment? And the last question is related to the global warming. Do you believe that global warming is have a great influence on the diversity of the viruses in the field, and can they really give like an influence that someday they may make some virus become more pathogen and become like a emerging disease like a COVID-19 in the future? Thank you.
Those are all really good questions. Bagus. Thank you. Yeah, so I haven't presented the work that I'm working on now because I don't want to get in trouble with BRIN and CSIRO sharing it, but maybe I would have been fine. But currently I'm trying to use, well, we're using a couple of different methods, but one of the methods is metagenomics, maybe with the MinION, but a little bit with NextSeq, trying to identify viruses from environmental surveillance. It's the question. The first question was sort of like, what is the point? Is it better to use like some of the I forget what they're called, sort of like the COVID kits, but for detecting certain viruses, or should we just do PCRs? I think that's pretty valid. We are doing a lot of PCRs for our surveillance at the moment. It is quicker and easier to detect certain groups. The only issue is abundance and type of virus. So metagenomics allows you to find viruses that are a little bit more divergent and might be at low abundance. So these are like in organisms like bats or in bats and rats. There's a lot of the populations can hold a fair amount of virus diversity, and not all of it is very, very highly abundant. The idea of the metagenomics for this kind of thing is to find viruses that are, say, close to the coronaviridae or close to the paramyxoviridae, but not quite captured by your PCR. It lets us look at multiple virus groups at once, so we can see things that we may not be concerned about. So some of the samples I'm looking at orthomyxoviridae, which are influenza. I'm looking at paramyxoviridae, which are Henipa. I'm looking at coronaviruses. But without doing metagenomic sequencing, we might not know what kind of arenaviruses are in there. I think practically it's a pretty hard approach to make work for these sort of things, I think. Like practically, when you get down to it, you kind of want to do several PCRs for your specific groups. But I have a dream that if we could get it used for surveillance, if I can make it work for MinION, it could be quite a good approach, because we don't need to know what we're looking for beforehand. We can do it quickly in the field, and we can find any virus at all, which would be kind of neat. But there's a lot of hurdles to making that work at present. The price, the speed, actually making the bioinformatics and workflow work. That was a great question. Let me go to the second one. COVID has evolved genetic markers. Yeah, so both influenza and coronaviridae, or myxoviridae, sorry, there's lots and lots of specific genetic mutations that are predictive of, say, influenza becoming highly pathogenic, or coronaviruses becoming more dangerous in certain ways. And you can, though, seeing those mutations does sort of allow you to predict a bit. And I think I said this way at the start, the projects that I'm working on now are more about preparedness rather than predicting and prevention, because I believe like you can sort of predict based on mutations. If you find a strain that has this mutation, you go, okay, that's pretty likely to be pathogenic, or it only needs a couple more of different mutations that we've seen. But there's two things that make it, I don't think, a great approach to be trying to do prediction as such. And one is epidemiology, like the population structures and the way that the viruses move are very, very important. And just looking at the genome doesn't really capture that. So viruses that became epidemics, so coronaviruses spilled over into humans 7 times before the actual coronavirus outbreak. The key part of the coronavirus becoming emerging infectious disease is that crossing from wild host into domestic, or wild host into humans. And it's very hard to, from my side of things, find viruses in environmental samples in bats and then say for certain, based on these genetic markers, that this is a virus that'll cross over, or this is a virus that won't cross over. It's very determined by epidemiology and the opportunity. You know, a lot of emerging infectious viruses came from repeated spillover events, where viruses are coming into contact in interface, they're spilling into a domestic animal or into a human, they're doing it again and again and again and again, but they're not causing outbreaks, or they cause minor outbreaks, they don't cause epidemics. During those spillovers, they might acquire mutations that we could maybe work out what the reason, the mutations that we could go, okay, this is what's going to cause it, pathogenesis, but it's going to be different for different hosts, different host pairs, and it's quite, it's the epidemiology of the situation. I know I shouldn't say this because I'm bioinformatics and all my focus is genomes, but it's the epidemiology that is the big unknown bit for whether a virus is going to emerge and become an outbreak, I think. But in existing outbreaks within the coronavirus, within influenza, we can look at genetic mutations. But again, it's been very well known to be unpredictable. It's quite hard. Again, I think epidemiology, you know, viruses crossing into immune compromised hosts, crossing out into minks or into ferrets or viverids, and then coming back, or into birds and then back again. A lot of those dynamics go towards whether a virus will be pathogenic or spread faster or acquire certain mutations rather than what the mutations are themselves. That was question 2. I love that question. Fun question. Global warming. Oh, this is also a good question. So does global warming affect the a presence and danger of RNA viruses in environments? I think this is a massive, this is, I believe that is true. And it's a huge topic. I think it's probably very relevant in Indonesia, actually. But I've looked at pathogens in China, and I think they're similar to Indonesia in that the wildlife here, there's so much diversity, right? There's a massive diversity of mammals and different organisms in rainforests and forests in China and in Indonesia. With global warming and clearing, these habitats change so that these organisms can, again, it's not exactly a genetic thing. It's the interfaces, it's the spillover, it's the epidemiology of the situation. With global warming and clearing, humans and domestic animals are more likely to come into contact with wildlife. You change the wildlife distribution, the higher chance we have more contact. And I think for an outbreak, a zoonotic to occur, you need this recurring spillover, and you need to look at this evolution at the interface. And that's part of the logic of this project. I guess whether it's caused by global warming and clearing, I believe so, but it's I guess my project is a bit more applied now. But yeah, we want to really target these interfaces where due to global warming or due to clearing, that distributions of animals have changed. Just to clarify, I mean, distributions of animals change due to global warming because of temperatures. It can change where animals are used to a certain temperature. Their populations will move as the temperature changes over the country. Or if you have in, say, Indonesia, less and less rain events or more rain events, more category 2 cyclones, this can change the distribution of your organisms, maybe allowing more fish to spill into areas where they're not usually there with higher rainfall. But it's a complicated topic. But yeah, I definitely think global warming and urbanization of some areas very directly affects how often we're going to have outbreaks and spillover events. Thank you, Riza. Sorry for the long answers, if that's what.
Can I ask you a last question here?
Sure.
I want to get your opinion about the COVID-19. Is it really a cure by nature or man-made as a global conspiracy?
Oh, yeah.
Because it's too good to be true that it occurred by nature, because it's so suddenly. So I know I want to know your own opinion as a scientist here.
Yeah.
And
I wonder if I can. Yep.
And more question that is there any politic in the outbreak of virus, because it's weird to me that they changed name from Wuhan virus into the COVID-19. So I think it's like a political here play in the scientific area here. So I want to know your opinion about this. Thank you.
Yeah, so much politics with this kind of stuff. I've worked a lot with research in China, and it can be very complicated. But also just research involving most governments are fairly complicated. Let me share a slide with you. This is from another talk I did like last week, I think. It was just for the public, so it's not exactly aimed at scientists. I will find the slide that I want to talk about. Is this the right one? It's a little similar to the talk we just did. No. Yes? Yes. Can everyone see that? Mostly showing it for Riza. There's sort of 3 hypotheses for the origins of coronavirus, so COVID, the pandemic. That is, it was passaged in a lab. So the 2 of them are whether it's passaged in a lab, 1 as use as a weapon, or 2 as used for research. In a research capacity, they're passaging viruses. The 3rd option is it's from a natural host. Obviously, I followed a lot of this fairly closely. The argument against it being from actually a developed virus, like man-made for the purpose of making an outbreak or something like that. The argument against that is that there's actually a couple of different arguments, but the main one I think is that the specific genetic changes, so the changes to I think it's the furin cleavage site in the spike protein, are not anything that we'd seen before this. They're not like well-known naturally occurring genetic changes. They're not genetic changes that you would expect to see if someone made this on purpose. There's no artifacts of genetic manipulation. So the idea is if someone, it would be astronomically hard to make a virus that had these genomic changes, because these are genomic changes you hadn't seen, and it would be very hard to purposely make that happen. So there's people who can make a lot better argument about the origins of RNA of coronaviridae. I think the coronavirus SARS-CoV panic. I wonder if I don't have any paper references for it. But I showed this slide because the man-made, purpose-made virus theory, I think it doesn't hold a lot of weight, and the consensus is it's probably not the case. There's a little bit, it's a little bit open to maybe there could be a lab that was passaging the virus, but even then, the main theory at the moment is it's host associated. So this tree here, I think we have oh no, it's not a very good tree. Do I have a better tree somewhere? I don't really. So SARS-2 we can see SARS-CoV-2. I'd really like a pointer here, but a lot of the closest relatives that they found were bats and then pangolin, which is kind of weird. So a lot of the closest relatives are these bat viruses. This paper that I have on the slide was an attempt to try and like geospatially analyze, try and work out exactly where this where the start of the outbreak was. And I just think this is really cool, but I guess it's sort of also an argument for a host origin for COVID. And that is, this study looked at the time that detections were made within Wuhan, and they went backwards. So they went backwards in time over the appearance of the infection, and narrowed it down within the city where they think it would be, which is this Wuhan seafood wholesale market, which is sort of the market that a lot of people suspected would be the origins of COVID, to the point that they did a World Health Organization mission where they went to the market, and that's what this map is, and swabbed all across the different cages. They did PCR and detected how much COVID they could detect, and the colors there, amount of environmental positives, the amount of positive swabs through the PCR that they found within the market. I showed this talk. This talk was for the general public. It's like a citizen science thing, but I showed this because I thought it was really neat that they could narrow it down to a single cage. And then there was photos taken by Eddie Holmes a couple of years ago before the pandemic, showing some of the animals in the cages at that stall in this market, which I think is crazy. The evidence overall, I'm not sure if I'm making very good argument, because I'm not an absolute expert in this, and there's definitely some people or papers you can read to really learn a lot about it. And there was a massive debate, right? But the main going theory is that there was some kind of host organism. They don't believe it could be bats, but I think the theory is that it went from bats to some kind of mammal and then to humans. These cages had birds, but they also had these raccoon dogs, which I think is my favorite COVID origin host animal, I think, because of the story. Yeah, kind of tanuki looking. Yeah, I don't know where I'm going with this now, Riza. My opinion is it's animal origins, but the animal origins is very, very cloudy, because there's a lot of obstruction to finding out what exactly happened. There's undersampling. There's a lot of groups that I want to say it's this or that. I know in America there's like tribunals about whether it was lab made or not. But yeah, I won't get too more into it. I just think it's an interesting topic. Thank you, Riza. I'll check with.
Ada lagi Bapak Ibu?
All right, I'll hand it back to Sugi and see if there's any other further questions.
Yep. Thanks, Callum and Pak Riza, for your questions. Are there any more questions to Callum related to RNA viruses and from environmental samples, especially? Anyone?
Ruby?
Is Ruby there?
Yes.
Okay.
Yes. Thank you. Thank you, Callum. Such a nice presentation. I have a question about the archaeal virus, especially in hypersaline. When you find part of arenavirus in hydrothermal environment, but there's none of the RNA virus in the hypercell. What is your hypothesis for the condition on the hypercell lake? Is the how to say this, the stability of the RNA capsid or anything else?
So that would be what the reason I think for finding none was? Did I get that right, Ruby? Okay. Yeah, my favorite theory, and the one I want to be true, is that there's a group of RNA viruses infecting these organisms, but they're so ancient and so different that it's very hard to find them through sequence alone. But and I really like this theory. I can't say if it's exactly true or not, but there's a theory that some of the first organisms before the last universal common ancestor were replicative RNA. And the idea is that RNA viruses all of those trees are one big tree, and the origin is right up just before the first bacteria archaea, right? And because of that, RNA viruses have evolved since then in parallel to all organisms. So part of the looking for RNA viruses in archaea was I thought it would be, I could find evidence for this theory if I found some RNA viruses in there. Because we didn't find any, I think some of the alternative hypothesis, and I probably I spoke a little bit about it, but some of the alternative hypothesis is that just the structure of replicating RNA is just not stable enough. We see so much DNA virus, but no RNA virus. It could be just that the structure of RNA viruses are not built for these kind of environments. That would fit more with a different theory that a lot of viruses in general might have arose from genomes. So there's no ancient origin. It's just that elements of host genomes from microbes or something leave the microbe themselves and become mobile elements replicating by themselves. This would make more sense with that kind of theory. Because if there was an RNA virus origin for everything, then you would expect RNA viruses to be everywhere and to be able to cope in these environments because early Earth environments were like this. But yeah, it could be that RNA viruses just are unable to live in these environments or the cost is too high. Interestingly, I think there's a lot of differences, like different types of environments that archaea live in. So that it would be that RNA viruses can't cope with any of these types of environments: lack of oxygen, too much salt, high acidity, very high temperatures. The third thing is if there's some sort of bias in the way that we're looking, so sequencing or metagenomics, that makes us unable to see the RNA viruses that are in these samples. There's some, maybe because of clay content, maybe because of the samples interfering with our workflow, it's biased against seeing these RNA virus types, in addition to them being quite divergent. Did that answer the question, Ruby, or is that all over the place? Okay.
Sorry. What is the follow-up question? What's your suggestion if someone wants to dig up more in this RNA virus, especially in the hypercell lakes?
Yeah. 2 or 3 things, I think. Well, yeah, I think like with my study, finding a negative result that's very, very valid is like that you can be certain is true is a really meaningful result, right? Because there's different theories about how viruses arose generally. It's fundamental research, but there's theories on how viruses arose generally that could be answered by whether we can find or add evidence to if we can find RNA viruses in archaea or not. So finding none, I think, is kind of an interesting result in itself. A whole kingdom of life, no RNA viruses. Approaches, I think, to looking at this sort of thing would be, one, you want to you want to try and have as pure archaea cultures as you can, similar to what I did, but maybe even purer. Maybe enrichments are probably the best way to go. Because if you find an RNA virus, you could put so much work in, but then it'd be a major pain if you have some other bacteria or eukaryotes in there and you can't conclusively prove it. So I'd recommend very high large components of archaea if you're going to have a go at it. The second thing is utilize the stuff that I didn't utilize. So there are some AI virus detection, RNA virus detection tools out there. And as well as that, there is AlphaFold, which I didn't use sufficiently. So the idea that I'd have is information is more conserved as you go along that dogma. So DNA and RNA is the first level, but because 3 nucleotides code for 1 codon, and you can change some of the nucleotides and you still have the same codon, there's redundancy. More information is conserved in a protein sequence over a very long time. So you can have virus genomes that look really different, but their protein, like you can't see the similarity, but their protein sequences are quite similar, or in a way, some similarity there. The next level up from that is like the way that the proteins fold. So you could have sequences where the proteins aren't sequence, the amino acid sequence is different and you can't find similarity, but the folding, like the actual structure of the protein, you can find similarities. If I had more time, this is probably the approach I would have tried to keep going with, but it took a lot of time to get where I did, really. But yeah, the second thing, apart from try and getting as pure as possible cultures of archaea, would be to use AlphaFold to predict structures and then try and find structural similarity to viruses. So if they are there, RNA viruses are there and they're very divergent, we can't detect it with the tools that we have, I think trying to find structural similarity would be the way to go. Aside from, there is some AI tools, like machine learning tools that have been trained on to detect RNA viruses. But that AlphaFold thing, it's a little tricky to say once you find a sequence where the AI tool goes, this is it, you've got to work out why, and then you've got to annotate it, which is a bit of a tricky job. Yeah, that's my opinion on how to keep going with the archaea RNA viruses, I think.
Thank you.
No worries.
Right, thank you, Ruby and Callum. Anyone would like to ask a question to Callum? Yep, I think we need to the end of this webinar, but I have one more question to Callum. Yep. In terms of like revealing the biodiversity of virus in environmental samples, how to correlate your findings to the evolution or the viral circulation? Did you perform like a longitudinal study, or maybe just compare with the previous database of virus?
Yeah. The stuff I did in my PhD was fundamental research. So the application is very long-term or pretty limited. The funding was more for discovery and finding new information, research for the sake of research in a way. Some of the applications though was, like I said, we can't detect viruses that we don't have any similarity to. So the further out we can find genomes for viruses, the more viruses we can detect. And that can be useful in that there could be groups that we just don't know about that. I don't exactly believe this, but there could be, well, maybe for animals, there could be groups of viruses out there that we don't really know or understand that could be at risk for agricultural animals or plants. Maybe not humans exactly, but we see this with some of the big, like at one stage we thought influenza is just human specific, or orthomyxos infect just certain groups of animals, but they found, orthomyxos in insects, in fish. These groups are much larger than we previously thought. And I think uncovering more of these groups could show ones that are far more ubiquitous. But yeah, the research in my PhD was fundamental research. So the funding is kind of a difficult kind of funding to get, I think. The other thing I was going to say is, yeah, it's just sort of broad. There's definitely things information you can find from looking at these viruses for certain technology that might be useful. But yeah, it's very blue sky research, fundamental research, this one. Pretty different from what I'm doing now, but some of the skills apply.
Yep. Oh, okay. Okay, Callum, thank you.
Sama-sama.
Terima kasih. All right. I think there will be no other question from this. So once again, thank you, Callum, for your talks, and that's been fantastic and give us more understanding and knowledge about viruses from environmental samples. And then, yeah, I'm happy to have a collaboration with your team as well to study this. Yep. Okay, and then I'll give it back to Ms. Lucy. Thank you.
Okay, thank you, Mr. Sugiyono, for leading a very interactive discussion, and thank you, Dr. Callum Le Lay, for a very interesting presentation. And as a token of our appreciation, we will give a certificate for Dr. Callum Le Lay as a speaker. Thank you very much for sharing your valuable knowledge. Ladies and gentlemen, this is the end of our agenda for today. I'm Lucy Arini. On behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. See you in the next event. Wassalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Callum.
Assalamualaikum warahmatullahi wabarakatuh.
Thank you.
Thank you. Terima kasih, Callum.